The most economically devastating viral disease to the swine industry is the porcine reproductive and respiratory syndrome virus known as PRRS. PRRS virus replicates in the pulmonary alveolar macrophages and induces prolonged viremia that lasts for months. This predisposes these pigs to other bacterial and viral pathogens, and to date, vaccinations have not been effective. So the only way to eradicate the PRRS virus from your herd is to euthanize the entire herd. Symptoms in gilts and sows are reproductive failure with an increased rate of late-term abortions, early farrowing with a decreased litter size and an increased number of mummies. In boars, there's decreased libido, fever, and a decreased sperm count. And then in young and growing piglets, we have cases of pneumonia, diarrhea, and a mortality of 12 to 15%. Financially, PRRS costs about $660 million annually in North America and about 1.5 billion euros in Europe. This is estimated to be about $6 million a day. There's a cost on our sustainability. The amount of money that goes to produce these pigs is just lost. The threat to our food security, when entire herds are being euthanized, what is our food source going to be? As well as animal welfare. These animals get very sick. Not to mention the emotional toll that goes on for the farmers and their families when their entire lives are put into producing these animals and it's just lost. So the proposed mechanism of infectivity is a close association with the heparin sulfate on macrophages, and then binding and internalization by a protein called sialoadhesin, and then CD163 is thought to play a role in unpackaging this viral genome and depositing it in the cytoplasm to allow for replication. Siglec-1 has thought to be the receptor. However, we have created Siglec knockout pigs, and these pigs are still able to be infected by PRRS, so we have now moved on to CD163 knockouts. We've created CD163 knockouts by two different targeting approaches. First, CRISPR-Cas9 mediated editing in fetal fibroblast cells, followed by somatic cell nuclear transfer, and then injection of CRISPR and Cas9 directly into zygotes. We have created multiple different litters and have done multiple challenges. However, I'm just going to be talking about our very initial challenge today, in which we looked at 3 CD163 knockout piglets and 8 CD163 wild-type pigs. They were randomly assigned ear tags, and they were shipped to Kansas State University, where they were housed in a BSL-2 large animal facility. They were allowed to acclimate for 3 days, and then they were challenged with a North American PRRS isolate. The pigs were maintained in the same pen so that there was constant contact between all of the pigs. And we monitored these pigs daily. The study was then terminated 35 days after infection. So looking at the clinical signs that we've seen in these pigs, in our wild-type pigs, we see respiratory distress and fever, but in our CD163 knockout pigs, we did not detect any respiratory signs or fever. Then we wanted to look at the level of viral RNA. This is a typical viremia curve of pigs that are infected with the PRRS virus, where viremia levels peak between days 3 and 14. Kansas State University has challenged over 3,000 pigs, and this is what they see every single time. It was not until they challenged the CD163 knockout pigs that they did not detect any viral RNA. We also looked at antibody production, and as you can see in our wild-type pigs, we have high levels of antibody production, but none detected in our knockouts. So where do we go from here? For our CD163 pigs, they're being monitored to show that there's no unintended consequences on growth, feed efficiency, or health. Genus has licensed this technology and is looking for FDA approval. But what other edits can we make for production agriculture? Are there other disease-resistant swine models that we can produce, such as the African swine fever, swine influenza virus, and the porcine delta coronavirus, which has just recently been shown that it may actually be able to infect humans as well. So we have found that by using CRISPRs, we're able to answer both basic and applied questions about domestic animal biology. And with that, I'd like to thank you.